Okay, so this is our Lab 11 review. So this is our last lab of Block 2. A lot of these things we've seen before, just now we're looking at them in a new context, or at least with some context. So first we have our parahippocampal gyrus, and that is going to be the most medial gyrus on the bottom side of our temporal lobe. So you can see that right here, specifically this folding. And it's not just this guy right here, because this is actually our uncus, and it looks like it's almost a separate little gyrus because of how deep this is. But this little streak is actually just a really well-defined intrarhinal sulcus. So basically your parahippocampal gyrus is one sort of whole being, and then the uncus is that bump that we know of that sticks off, and it is delineated. It's still part of the parahippocampal gyrus, but this little folding that separates it is that intrarhinal sulcus. And then another gyrus we've seen before is the cingulate gyrus, and so here's our corpus callosum. Our cingulate gyrus is that gyrus overlying it. So this is that cingulate gyrus. Deep to that, those axon fiber tract, that's going to be the cingulum. And then also, let's look back down at that uncus here. Deep to the uncus is our amygdala. And then also just another limbic structure that we needed to have some housekeeping on is the mammillary body. So this is a fiber dissection, so you can see things look kind of weird around here because all of the flesh has been sort of chiseled out to get these fiber pathways pretty visible. But the mammillary body is actually going to be right here. And so the mammillary body has a connection with the hippocampus, to the parahippocampal gyrus, and the hippocampus via the fornix. And so you can actually see the fornix coming from mammillary body up, and then here's your fornix that's going to swoop around and end up down into that parahippocampal gyrus. Okay, so this is sort of like a Heimer dissection to the max, and basically we've just isolated the hippocampus out of the brain. So it's kind of hard to orient yourself to this structure, but here's anterior, posterior. These are going to be sort of parts of your temporal lobes. And you can see here, this entire little formation here is our hippocampus. And this is the best picture that I've seen of the pes hippocampi, which is the most anterior part of the hippocampus, looking like a cat's paw, like Dr. Smith says. So here are the little cat's paws. Those are your pes hippocampi, and then this whole little structure here is the hippocampal formation. Okay, so now taking a look at some cross sections, let's go ahead and orient ourselves with some stuff we already know. Let's look for our lateral ventricles here. Lateral ventricles, these are going to C-shape, posteriorly and then arch around into that temporal lobe. So here is that little hole delineating the temporal horn of the lateral ventricles. And here's the other side. And so what we already know is that our caudate nucleus does the same thing. Our caudate sees back and then comes, and actually the tail of the caudate becomes the roof of the temporal horn. But now what we're learning that's new is sort of that the hippocampus is becoming the floor of the temporal horn of the lateral ventricle. And the hippocampus is sort of, the parahippocampal gyrus is folding in on itself and sort of spiraling. And so the spiral has different parts that have different names. So the part that's the most inferior, so like just starting to get curved into this curve, is the subiculum. So sub, beneath, so subiculum is this portion that's like folding in the first. And then the subiculum transitions into the hippocampus proper, or the hippocampal gyrus, and this is sort of this arch up and around here. And then it ends in the middle as the dentate gyrus. So the dentate, as Dr. Brewer said, is the middle of this hippocampus jelly donut. And so we can look over here closer. This is going to be our screen left side of the brain. So we have our subiculum folding up into our hippocampus proper, and then our middle little dentate gyrus right here. And now some parts that are sort of part of the hippocampus but are also named as parts of the fornix, which we'll look at in a minute, are the alveus and fimbria. And so the alveus and fimbria are axon fibers, so you'll see them stained darkly here. And basically, it's coming from the subiculum and perihippocampal hippocampal gyrus, and it's these black axon fibers here arching up over top. So this little thin line is the alveus, and then the alveus you can see thickens, and this little bulb that it sort of thickens into is called the fimbria. So on the side too, you have your alveus and then your fimbria. Okay, so now we're looking at our inferior view up at the brain. So let's again point out our parahippocampal gyrus. Like I said, it is that medial most from the inferior view temporal lobe gyrus. So here's your temporal lobe. So let's look most medial. We have this gyrus right here. And you can see your uncus really well, this bump right there, and then the little line of your intrarhinal sulcus. Now there are also a couple more regions of this parahippocampal gyrus that now we can see. So I'm going to hop over to this image because we have got it zoomed in. So uncus, parahippocampal gyrus. So your entorhinal cortex is going to be basically lateral to the uncus. So here's your uncus. So then this area is going to be your entorhinal cortex. And this cortex is actually one of the main afferents into the hippocampal formation. So it's going to submit information to the subiculum, and then it's going to take it through that pathway, and it's also going to help consolidate memories. It's going to shoot back to the CA1 area to sort of consolidate memories, the end of that circular pathway. It'll sort of shoot back to the end and like basically tie up some strings. So entorhinal cortex. Then you have your piriform cortex, and we've heard this because we remember the piriform cortex is that primary olfactory cortex. And so the piriform cortex is the anterior most tip of the parahippocampal gyrus. So it's going to be right here. This area is your piriform cortex. So you'll have them here. And then your entorhinal cortexes. Uncus, amygdala is deep to that. And then I just have listed here the hippocampus afferents. So you have a lot coming from the cingulate gyrus. You have a lot coming from the parahippocampal gyrus, specifically, like I said, the entorhinal cortex. And then you also do have some coming in from the amygdala. Okay, so this is a Fornix slide. I've got a lot of different ways to look at it because it's kind of confusing. It's kind of C-shaped, just like a lot of the other things in the brain are. So the Fornix, like I said earlier, it connects the mammillary bodies to the hippocampus. So you have mammillary body, Fornix, hippocampus. So it's just a fiber pathway. So it's not too complicated, but it has three named portions of it. So we have the body is just the main stretching anterior to posterior area. Then they come down in the front and turn inferiorly, and these are the columns, and they're kind of close together here. They V off and separate into these columns towards the front. And then they do the same thing. They V off and come down towards the temporal lobes, and these are the crus of the, you can't see the other side, obviously, but these are the crus of the Fornix. And then again, I pointed out the alveus. So the alveus, because we really pointed it out on the hippocampus, it's going to be like these fibers here, the alveus thinning, and then it thickens here and sits on top of the hippocampus as the fimbria. And then just a little FYI, the alveus is composed, those axon fibers are being made from, like I said, the subiculum and the hippocampus proper or hippocampus gyrus fibers. And then the Fornix is actually suspended it's hanging up from the Septum Pellucidum. So here's your Septum Pellucidum. In this gross image, the Septum Pellucidum has been carved out, but you see that this is what was holding the Fornix up from the corpus callosum via the Septum Pellucidum. And then also the hippocampal commissure is something we've had to point out a few times, but it's really hard to see because it's not very well developed in humans. But the hippocampal commissure, if you more just memorize the location of where it's supposed to be, that's really what you need. So the place where you're going to find the commissure of the hippocampi is going to be on the Fornix, and it's going to be right basically at the border between the body and the crus, so at this bend. But the best way to remember that is that bend is basically on the anterior border of the splenium of the corpus callosum. So literally right here is going to be that hippocampal commissure. And yes, like I said, it lines the splenium. Okay, so now we have some more cross sections, our best friends. We've seen these a billion times, but now we're going to point out something new on them, the fornix. So here we can see, are we really anterior or are we really posterior? Okay, so I'm seeing a really big pineal gland, posterior commissure, big red nuclei. This looks like midbrain right here, or brainstem, I mean. So we're pretty posterior. And we see where we have our lateral ventricles. So here's our corpus callosum. And we see these little things V'ing out really far apart. This is going to be our crus. So when we're naming parts of the fornix, it's really just taking into account how far forward or back you are in the brain, because you know up in the front, you're going to have your fornix separate into columns. One way to distinguish those is just, are you far forward or are you far back? But also in the back, they're going to V out way wider because they're trying to end up reaching that temporal horn. But in the front, the columns are just V'ing apart. So here's super anterior, or, well, you know, not super anterior, but anterior enough that we still have a really good view of our optic tracts, and then we're just at the beginning of our thalamus here. You can see our columns of our fornix coming down right here. And then we actually still see a bit of our septum pellucidum hanging that up. And then our columns are just V'ing out. And then our body, I have a slide coming up of our body that shows it just kind of looks, you know, a lot flatter, like you're not seeing that V, you're not seeing the separation quite as much. I'll show you in a couple slides. But let's see what else I need to point out here before we move on. Oh yeah, I'll just point out the alveus and fimbria again. So we have the black lining over the top of the hippocampal formation. That's the alveus, thickening into the fimbria. Same over here, alveus, fimbria. And then a little throwback, we've got our subiculum, hippocampus proper, dentate. And then if we look over here on our little anterior view here, we have our foramen of Monroe. So we're pretty far anterior. Here is our anterior nucleus. Here's our dorsomedial nucleus. And this is probably anterior enough, it may be that this is our VA. If not, VA, VL is good enough to distinguish those. Here's a good view of our cingulate gyrus and our cingulum, those black fibers within that. And then let's look back over here. We have our MGN and our LGN. Any of these black fibers sort of going up to that LGN is going to be our optic tract, because like we know, the optic tract is sending its fibers to that LGN. And then to throw back to the last lab is that we have our thalamostriate vein right here, that lumen. Over top of that is going to be our stria terminalis, and then this little groove is our thalamostriate groove. Okay. So here's some more fornix. Let's look where we are in the brain. I immediately see the big anterior commissure because we have this A, like we learned. The A is coming down, and what lines it is that anterior commissure. Well, now to add to that little way to remember, these parts of the A coming down are actually our fornical columns. So those are going to be the columns right there. Here's our septum pellucidum holding it up. And other ways, just to solidify that we're anterior, is we don't have the thalamus showing yet. This is our caudate head. And so this means that this is our anterior limb of our internal capsule. And below here looks like this is going to be our putamen and our globus pallidus externa, but not necessarily vital for this exam. Moving over here, this is what I was talking about, the fornix body. These are really close together. They're not really V'ing off anywhere. We're just kind of following this straight back through the brain. So this is when it's taking its long anterior to posterior path side by side. So here's the lateral ventricles, corpus callosum, cingulate gyrus is cut off a little bit, cingulum within that. We have a good view of our mammillary bodies, so we know we're sort of like, we're somewhere in the region of not super anterior or super posterior. The mammillary bodies aren't necessarily in the middle of the brain from anterior to posterior, but I always imagine, okay, I'm somewhere around the middle-ish. But also, check this out. So our mammillary bodies we have a pretty good view of. We also have a pretty good view of this structure right here. Now, this little line, you can see just barely connecting our mammillary body all the way back to this nucleus. So the trouble with identifying this little track right here is that when we see the mammillary body and we see something coming off of it, it's probably going to be our mammillothalamic tract. I'd say most of the time, because it's mammillary body to the thalamus, and the mammillothalamic tract is connecting that mammillary body to the anterior nucleus. But I did make a really good point, which is still true, that when you are cut straight through the mammillary bodies, like once you get to the mammillary bodies, this top nucleus you're going to see on the thalamus is actually the dorsolateral nucleus because we've gone past the anterior nucleus. But this may be an exception, not an exception per se, but because you can see, I think that this cross-section has been taken like when they just barely have scraped into this mammillary body. Like I think that there's a lot of mammillary body left behind this cross-section. So that said, we are right on that transitional zone between anterior and dorsolateral. And because we see this perfect tract, I'm going to have to assume that this is the mammillothalamic tract and this is that anterior nucleus. But then there's also this internal medullary lamina that is going to occupy essentially the same space because they sort of actually end up running together. Like the mammillothalamic tract is going to use that internal medullary lamina as like a highway to get to that nucleus. So I don't think that we'll be asked to differentiate between them because I think that's a little mean, but a way to tell the difference per Dr. Smith would be if it was going to be our internal medullary lamina, it would be a lot thicker and you would really see it grabbing the sides here. Like you would be able to delineate the Y shape a lot more. So I think that this is just going to be our mammillothalamic tract. Okay, so let's orient ourselves as we always do. I'm immediately saying anterior because here's our nucleus accumbens. That's my go-to. When I see owl eyes, this looks like an owl eye with its little eyelashes here. That means we're super anterior because we are so anterior that the putamen is reaching around and meeting via the nucleus accumbens that caudate nucleus. And so that is what I'm looking for. Now, granted, since we're so far anterior, we have a new little player we want to take a look at. So we have our septum pellucidum, and then we would have our fornix, but we're actually a little bit in front of the fornix. And although these look a lot like fornical columns, the best way to differentiate these columns from this being the septum pellucidum coming down into these septal nuclei, which is what this is. There are two ways. First of all, we have these lumen right here. These are our septal veins. So meaning this would be still part of the septum, but also our septal nuclei and that formation here is completely continuous with the nucleus accumbens. So like you can see here, it's not like it's sitting up against it. It is literally a part of it. Our fornix is not going to be grabbing, it's not going to be molding into any structure like that until it gets down to the mammillary bodies. So when you see something continuous up towards the front and you're like, okay, is it fornix or is it septum? It's going to be septum. So like I said, here's where your septal nuclei are going to sit. They're kind of thickening out towards the bottom for these nuclei. There's your septal vein lumens. Here's your thalamostriate vein lumens. And then our fornix actually does project to the septal nuclei, and it's going to be sort of posterior to this formation. It's going to be projecting to these. And then also, since we are anterior, this is our anterior limb of our internal capsule. And then since this was our thalamostriate vein, here's our caudate. Over top of that vein, again, is the stria terminalis. And then this is going to be our thalamostriate groove location. And then also, we can see these black fibers going underneath the lentiform nucleus, aka putamen, and then our nucleus accumbens. This is going to be our sublenticular limb of our internal capsule, because this is our lenticular nucleus per se, and so then these fibers underneath are sublenticular. So here we have our basal forebrain. This is an area more just a theoretical area than a place to easily point out. It's about the same location as the anterior perforated substance. So you remember that from like lab 1. It's on either side of the optic chiasm in these little pockets right here. So this sort of whole location is your basal forebrain, and that's going to be a big part of the cholinergic system, because we've got our basal nuclei of Maynard that are going to send some acetylcholine. So if we look from the side, it's going to be about this area. So we're seeing the corpus callosum sort of beneath that. And then here's our mammillary bodies just sort of above. So just this general location, and the basal nucleus of Maynard, I don't think we'll have to ID very specifically, but more just the general location. Then the ventral pallidum is like a delineation of the basal forebrain. And the ventral pallidum is specifically just the most dorsolateral region of the basal forebrain. So if we imagined the dorsolateral, so like the sticking out, there'd be this part of it. Here we would see like essentially this part of the basal forebrain would be our ventral pallidum. Okay, and then our amygdala. So our amygdala is, like I said earlier, deep to the uncus. It also is right on the tip of our hippocampus. So the best way to see it would be to point it out on a gross specimen. So let's look here. We have our anterior commissure, our fornical columns. So typically, when we look down here to our parahippocampal gyrus, we would be seeing our hippocampus. However, this is like the one time where that's not necessarily the case, because we are so far anterior that we are actually cross-sectioning basically right here. So we are hitting through that amygdala. We haven't reached the hippocampus yet. You can tell because you see just like this is a lot of gray, and you don't see that little infolding where you could point out the subiculum and the dentate gyrus. There's no little spiral. This is literally this is the amygdala proper. Like this whole thing is just the amygdala here and here. And I think that might be one of the only times. I think what we're more likely to be tested on is sort of asking what's deep to the uncus, and you can also kind of see the uncus bump right here. That's another good way to point that out to yourself. But I think it's more important just to realize that it's deep to the uncus and be able to point that out on a whole specimen, and just know that typically our hippocampus is going to be right here. But if we're anterior enough, look and see if we can point out the different convolutions of the hippocampus. If not, we're probably anterior enough where we can see the amygdala. Okay, so here's our lateral olfactory stria, and so this is pretty much was thrown in at the end of the lab, which is kind of funny. Like, it seemed like it didn't really fit, but it's kind of a throwback as well because we've seen this a billion times. So we have our olfactory bulb and our olfactory tract, and we remember from our last exam that it actually separated into two little pieces on this tract. We had our lateral olfactory stria and our medial olfactory stria. So you can really see that here. So here's our tract, and we see this little V. So our medial olfactory stria is going to be crossing actually the anterior commissure to trade sensation with the other side olfactory stria in that anterior olfactory nucleus. But the lateral olfactory stria was actually where that smell was going to be transmitted to the primary olfactory cortex, which we remember is that piriform cortex. And where is the piriform cortex? It is that anterior-most tip of the parahippocampal gyrus. So here's that gyrus, here's our uncus, here's our piriform cortex, and look, our lateral olfactory stria is headed right for it. Literally going straight towards it. And then here we can also see the same. So here's our medial olfactory stria out of sight beneath the chiasm, and then here's our lateral coming straight towards this piriform cortex. And here's our uncus, here's our entorhinal cortex, here's our intrarhinal sulcus. And yeah, you guys are totally ready to crush the practical. And I hope everybody does super well, and I hope we're all excited for spring break.